Hello, welcome back to another review of the MCAT biology section. Hopefully most of you guys already watched my previous episode. My first part going over just simple stuff. We went over cell theory. We went into the nucleus, kind of just explaining the nuclear envelope, and this is primarily for the eukaryotic cell. We went into the mitochondria, went over the parts of it, what it does, what's special about it. And then we finished up with the lysosomes, kind of just an oddball that needs to be covered. Today, we are going to hopefully finish up the cell review of eukaryotic cell, finish all the organelles that the MCAT really focuses on. So let's go ahead and get started. We're going to start it off with one of my favorites, I would probably say, the endoplasmic reticulum. So, endoplasmic reticulum. So, and I'm probably just going to say ER from here on out. Endoplasmic reticulum is ER. There is two main types that we need to know of. There's going to be the smooth and rough endoplasmic reticulum. I don't know why I wrote it like this, but I'm going to go opposite way. I'm going to talk about the rough first. But before we get to that, what is the endoplasmic reticulum? I'm just going to say it is going to be interconnected membranes. And membranes are just like, the cell membrane basically, but just instead of the main cell, it's for each of the organelles. So it's the interconnected membranes, just a series of them that is connected to the nucleus. So if I remember, I'm going to try to kind of put a picture of the cell up in the editing, and you'll be able to see that the nucleus is this big ball, and around it is this endoplasmic reticulum, right? Because we're going to get into sort of the workflow of the cell and where things go throughout the cell, and it's important to know that the ER is connected to the nucleus, because a lot of the things that are coming from the nucleus, mainly the mRNA that's coming from the nucleus, is going to be going to the ER before it goes to other places, right? And that brings us to smooth versus rough, right? So what makes an ER rough versus smooth? Really easy. Rough starts with an R, so we are going to know that in the rough endoplasmic reticulum, we are going to have ribosomes. Quick review of what ribosomes are is protein synthesis, right? Synthesis. So it translates mRNA to proteins. Okay? Now, that's all you really need to know about the rough endoplasmic reticulum. Going to smooth. What do we think smooth? What is the differential between smooth and rough, right? If rough has ribosomes and the ribosomes make rough, smooth has no ribosomes. And if you think of protein and ribosomes and protein, just connect them. What is the opposite of protein? If you're thinking like a gym bro, what is the opposite of protein? Fats. What are fats? Lipids. So I see the smooth ER as lipid synthesis, and hopefully that thought bubble makes sense. But it doesn't just do lipid synthesis, it also detoxifies and helps with transport. And this transport is mainly just transporting things from the rough ER to the Golgi apparatus, which is a nice little segue into our next organelle, the Golgi apparatus. A lot of people like to call, and you probably heard this before, the Golgi apparatus as the Amazon warehouse. To me, I wasn't a big fan of that because I never really saw the Golgi apparatus or an Amazon warehouse as doing something the Golgi apparatus does, right? The Amazon warehouse isn't changing the product, it's just shipping it out, right? Which is a key part of the Golgi apparatus, but it's not all of the Golgi apparatus. So the Golgi apparatus is just going to be an organelle that has once again, we're just sticking to the membrane, but it has stacked membrane layers, and it kind of looks exactly like that. It just is like a whole bunch of pancakes stacked on top of each other. I don't know why I just thought of it like that, but that's just the way I thought of it. And the Golgi apparatus is basically the Amazon warehouse, if the Amazon warehouse helped change the products at all, right? So it's not just a center point where things come from the endoplasmic reticulum and then are brought out to other places. They're also changed. Inside the Golgi, the products coming in are changed before they're then shipped out, right? I don't see Amazon doing that much. That's why I didn't really like that sort of thought process. I see it more of just, you know, kind of like an Etsy maybe. I'm not sure if that would make sense to people. Maybe people just like to stick to the Amazon. That's just not the way I like to think, right? So I'm gonna kind of think of this as a little work path, right? We have things coming from the ER, the endoplasmic reticulum, and they're going to go into these vesicles. And these vesicles, I'm just going to do a little drawing just to help it stick in your mind a little bit. These vesicles are basically transport circles, right? It's like a little Uber for these products, right? And products, we're talking about proteins, lipids, whatever that the ER is making. And what the ER is making is, of course, what the nucleus encodes for. So these vesicles from the ER are going to that Golgi, right? And Golgi apparatus, it's just shorthand. And inside the Golgi, it's important to know that we are going to have modification. So at the Golgi, we have modification. This modification includes the addition of, and it's not all the time, it's not like every single time a product goes into the Golgi, it's getting this, but sometimes we have the addition of carbs, phosphates, and /or sulfates, right? So it's not all the time these are being added in, it just can be added in, right? And another thing that is important to know is inside the Golgi or at the Golgi, we are also adding on signal sequences. And hopefully you can kind of see my writing. I know it's kind of small and scribbly because I feel like I just want to move a little bit faster through the material, especially because this is probably a lot of review. But these signal sequences are basically just little tags along the product, and these tags tell the product where to go, right? Where in the cell, where outside the cell, just where do we go, right? So kind of like GPS for product. Just where is our product going? Is it going to the mitochondria? Is it going to a lysosome to be destroyed into nutrients? What is it going to, right? And after the Golgi, where are we going? We're going ourselves back into a vesicle. And from this vesicle, we are going to wherever that little signal sequence brings us, right? So I'm just gonna say wherever. Wherever. Could be absolutely anywhere in the cell or outside the cell, right? And it just kind of brought this up. Outside the cell, if something was to leave outside the cell, it can leave through something called, let me spell it right, exocytosis. What exocytosis is basically just the vesicle. If you think of this little round circle as a vesicle, it comes up into a membrane. Let's have my chin be the membrane. It comes up to the membrane, and then it kind of merges, and then like the product is just sort of shot out of the membrane, right? So I'm just gonna say exocytosis is the process by which product is secreted from cell. It just leaves the cell through the membrane to go on to do whatever it needs to, maybe talk to other cells. Who knows what that thing's doing? It's just running around, okay? And boom, Golgi's done. Not bad at all. We're just following one simple workflow path, and now we get a little change up, and what we're working with, we're just going to go ahead and change over to peroxisome. English is not my strong suit. Science is. So peroxisome very simply has hydrogen peroxide. Pretty easy to remember. Peroxide. And what this hydrogen peroxide does is break down long fatty acids, via, and we're going to talk about this later in biochemistry section, so later as in a while, but you can start reviewing it now, via beta oxidation, okay? And that's mainly all we really need to know about the peroxisome. Kind of similar almost to the lysosome that we were talking about previously with the breakdown of material. Just, it's more specifically for those long fatty acids, and it uses hydrogen peroxide, right? So a little bit different, but very, very similar, okay? And now we kind of get into the meat and potatoes of this video, because for some reason, the Kaplan books and MCAT in general just want to talk about the cytoskeleton. So, overall, a general gist of what the cytoskeleton is. Kind of like our skeleton. It is for structure plus maintaining shape. So we, as a human, need our skeleton to sort of hold out my arm and move my arm, move my fingers. I need the bones inside my skeleton to do that, right? If I didn't have bones, I would just be a blob just laying on the ground, right? So we need that skeleton in place to help sort of hold the structure of our body, right? And it also, the word they use, conduit. I ain't using conduit. I'm just going to say it provides a pathway. Now, this isn't all cytoskeletons that provide a pathway, just some that provide a pathway for transport around a cell. Okay, perfect. Okay. Hopefully I can fit it all on this one page. That'd be pretty epic. But there's three main types of cytoskeletons that the MCAT focuses on. And maybe that exists. I don't know if there's any others that exist past these three. But just going over these three, our first one is going to be microfilaments. What these microfilaments are, cytoskeleton, and it is made of actin. So microfilaments is actin, which is pretty dang solid. You can see it as like more rigid, I guess. And what microfilaments do, and let me make sure I keep the microfilaments in for the video, is it provides protection. And here, let me actually do this. I plan to do this from the start, but better late than never. So microfilaments, they provide the protection, and they utilize or use ATP for movement. I'm just gonna say with myosin. Okay. Another thing that the microfilaments are used for, and we'll see this in the next chapter, chapter 2, is for cytokinesis, which is a part of cell replication. And they microfilaments just create the cleavage furrow, where the cell kind of becomes a circle, goes from a circle to kind of like a peanut shape, like that. I don't know if you can really see that well on the video, it might be too small, but the microfilaments do that, right? And that is all you need to know about microfilaments. Going into the second one, we're talking about microtubules. Microtubules. So microtubules are going to be used of, and this one kind of makes sense. Tubulin. And a key thing to know is that tubulin is hollow. This actin, we saw it as a solid, rigid, full circle. Our tubulin is going to be hollow, and it's going to follow a specific structure that we'll see later. The microtubules that we're going to see later, that kind of further expands upon like a hollow sort of nature. These microtubules are this second little dot we talked about right here. They provide our pathways for certain proteins, and it mainly brings up, I'm gonna say this, kinesin and dynein. So it allows for those proteins to carry. What do these proteins carry? If you were to think about the movement of things, what have we talked about previously? This part that move things around, vesicles. Okay, and I know this was kind of a bit conjuncted, so let me say this. So our microtubules, made of tubulin, they provide sort of walking pathways for the kinesin and dynein to carry these vesicles, which bring up these different products and cellular components around the cell. Okay, hopefully that sort of clears things up, because I know it was a lot all at once. Okay, and we also have a key thing focusing on movement and the movement of the cell. Okay, we have two I want to say factors in movement, not factors, two types of microtubules in which they affect or introduce movement. They have cilia and flagella. Okay, so cilia, we have materials on moving on a surface of cell. So things just moving around on the surface of the cell, like the cell. So cilia is moving things around on the surface of the cell, kind of like, imagine, trying to think of a good example. Imagine you're bowling a bowling ball down a bowling lane, which is full of little long hairs that kind of direct that bowling ball to a strike every single time. That's what a cilia is kind of like. It's not a good picture in your head, but just what it looks like. Okay, and flagella is going to be the movement of the cell itself. So a key difference between those. Okay, and I'm running out of space, but what I was talking about initially with the structure of microtubules, it is going to be a 9+2 structure. And let me see where I can best fit this in. I'm going to fit it right here. We're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9. When I have 9, and each of these, I'm not even going to try to draw it, are tubulin, hollow tubes, right? We have 9 surrounding, I'm going to change it to a different color, 1 and 2. I don't know why it's called the 9+2 method, other than the fact that it's 9 and 2 in different sections. I don't really like the way that it's 9+2 because that just makes me think 11. So I would think like 11 little things in a circle. I don't know. But we have 9 tubulins outside in an outside ring, a hollow ring, and inside the middle of that ring is 2 extra tubulins. So a 9+2 structure, 11 total tubulins making it. But that's sort of the process for it. And that's all for microtubulins. So kind of briefly comparing these, we have 2 separate types of components. We have actin versus tubulin. Microfilaments, being a solid rod, are more for protection and like more heavy-duty things, like breaking the cell into 2. Microtubules are more, you know, agile. They help in movement and, you know, walking pathways, right? And they have a structure, a clear structure, the 9+2 structure, okay? Our third and final one, and the final topic of this video, is going to be intermediate filaments. And I don't know if I'll have space to write over here. Filaments. Good enough, hopefully you can see that well. I probably could have moved it over a little bit. But intermediate filaments are just the third type of cytoskeleton that we're talking about. And what they're made out of, because that's always what I'm going to talk about first, is different, right? It differs. It can be made of, for different cells, different functions, whatever a cell specifically needs. This intermediate filament is going to be made of a certain different type of protein that is then going to change its function. You know, we're going to have keratin. And what do you think about keratin? Does keratin ring a bell in anything? Does it ring a bell in anything? I don't know. Okay. We also have desmine. Desmine. I'm sorry. You're just gonna have to work with it. Vimentin and lamine. Lamines. If you like soccer, then you would like that joke. Okay. Basically, what these intermediate filaments are specifically for is a few things. We have cell to cell adhesion. Don't think I need to really explain that much, but just, you know, cells sticking together, right? Your skin sticking together. We also have structural rigidity. What a weird word. Rigidity. And it also anchors organelles. Okay, so briefly going over intermediate filaments again. It has different proteins that make these intermediate filaments. Each happen on purpose for a specific function within the cell. And then the main things that an intermediate filament does is cell-to-cell adhesion, so holding a group of cells together. Structural rigidity, so that's like inside of a cell. Think of intermediate filaments as, I'm actually going to draw this out because I feel it would help to kind of visualize it. This is our little cell. These intermediate filaments are kind of just like a Spider-Man costume effect. They're the ones kind of holding that structural rigidity of it and anchoring, you know, our organelles. Here's our little mitochondria right here. So it anchors them in place so your cell isn't always just, you know, wobbling. And when you jump and you jump up and jump down, not every single mitochondria and nucleus and everything in the body just goes and goes to your feet, right? It sort of anchors those organelles. And along these pathways, of course, you're going to have your, let me get the color, microtubules, which are going to be, like nice little pathways, walking pathways. So it's not all just intermediate filaments making that network inside the cell. It's also these other ones that are playing a part. And you, of course, have, some maybe microtubules on the outside working as, cilia or flagella, just, moving things around or moving the cell. And then, whoop-de-doop-de-doo, we have to undergo mitosis, holy smokes. Let's go ahead and start to develop this cleavage furrow right there. I don't know if you can see it well, but this right here is being caused by those microfilaments. Okay, so overall, those are our three. Hopefully it makes sense. And just a short review of this. This covers our eukaryotic organelles. We had today the endoplasmic reticulum. You have smooth versus rough. Rough has ribosomes, so it synthesizes proteins. Smooth has no ribosomes. It does lipid synthesis, detoxifies different toxins and poisons possibly in the cell, and it is helpful in transporting things out of the rough ER to the Golgi apparatus, right? Going into the Golgi apparatus is sort of the Etsy, Amazon mixture where it takes things in, changes them around a little bit, kicks them out, right? So things come from, and let me use this laser pointer so if you're looking at the video you can see. Things come from the ER, the endoplasmic reticulum, into these vesicles, which basically transport things around the cell, not just within the Golgi apparatus, hemisphere. It also transport things out of the cell, wherever, right? But these vesicles then transport everything to the Golgi apparatus, where it is modified with either carbs, phosphates, or sulfates, things being added on to the cell. And we also keyly add on the signal sequences. These signal sequences tell the cell, or tell the product in the vesicle, where are we going? Where are we headed to? Where are we shipping off to, right? And that, of course, brings us wherever we want within the cell. Okay, and that might even include exiting the cell through exocytosis. Peroxisome, basically the lysosome, but it uses hydrogen peroxide, and it breaks down these long fatty acids, which we'll probably talk about in, I'm assuming, biochem, which are just basically, long carbon chains, lipids, everything like that. And it does it via beta oxidation. Once again, we'll probably talk about that more in biochem. And then finally, our last organelle is the cytoskeleton. It works exactly like our skeleton, mainly for structure and maintaining the shape of the cell, and for our case, our body. And another side piece, which is mainly for these microtubules, is providing a pathway to transport around a cell, and in the case of this flagella right here, moving the cell around different things, right? Main things we have are microfilaments, microtubules, and intermediate filaments, right? And I didn't really talk about it much here, but you can kind of think of these as different sizes, if that kind of makes sense in your head. And I don't think the Kaplan book covered this, so just make sure that it's sort of make sure it's correct. That microfilaments are like the big ticket items. It's the biggest, the strongest. Then you have these intermediate filaments. Then you have the microtubules, right? So filaments, intermediate filaments, tubules. Microfilaments are made by actin, microtubules made by tubulin, and intermediate filaments are then made by those different ones, and I just listed a few here. What they each do: microfilaments provide protection, use ATP for movement and cytokinesis. Microtubules provide that pathway and movement of things in the cell or on the cell and the cell itself through either cilia or flagella. And finally, the intermediate filaments: cell-to-cell adhesion, combining two cells together, structural rigidity, and anchoring organelles within the cell. Okay. So with that comes the end of this specific part. Next part, we're going to talk about a weird little sidestep within this kaplan, but going over tissue formation. We're going to talk about the different types of tissues, mainly the ones that are focused on this chapter. Should be a relatively short section, not too crazy, but I'm not going to get over ahead of myself. Of course, once again, thank you so much for watching, and if you have any criticisms, any helpful tips or tricks for not only me, but maybe your other study partners, anything else around, of course, I want to make this a helpful community that empowers everybody here to become successful. So thank you so much, and I love you all, and peace.